In today's lecture, we're going to discuss blood. As always, these are the learning objectives for today's lecture, and you want to make sure you can execute these before you proceed to the next lecture. Let's go ahead and first talk about the composition of blood, then we'll talk about something called the formed elements. So how does blood fit into the big picture? Well, blood obviously is a major component of something called the circulatory system. And the circulatory system has two parts. It has the cardiovascular system, and it has something called the lymphatic system. The cardiovascular system, if we look at it, is something that is composed of the heart and blood vessels, and then the lymphatic system is something that brings fluid from the cells back into the cardiovascular system. Now, if we look at this individual, before we start talking about the cardiovascular system and the lymphatic system, I want you to think of what is wrong with this individual if we're talking about the anatomical position. So think about it a second. What's wrong with this person if we talk about the anatomical position? Okay, well, this person should be facing you, right? And their hands are in the incorrect position, right? Their palms should be facing you, so their thumb should be on the outside. Their thumb should be lateral. Also, you'll notice his or her feet are not pointing towards you. That's something else that's incorrect with the anatomical position. One thing you want to think of is, what is the right hand? What is the right hand? Remember, it's always the individual's right, so it's this hand I'm starring here. Okay, just a quick review on the anatomical position. Let's go and talk about the cardiovascular system a little bit here. So first we have something that is called the heart, right? And the heart is the pump that pumps blood throughout the body. Now once we have the heart, we have vessels that leave the heart, and these vessels are called arteries. These vessels are going to be thicker, and they're carrying blood throughout the body. As those vessels diverge and become a little smaller, then we have structures that are called capillaries. After that, then they finally reach the cells of your body. After the blood reaches the cells of the body, it delivers oxygen, nutrients, things of that nature. Then what will happen is all the fluids that accumulate in the cells will be returned to the cardiovascular system via the lymphatic system. And the lymphatic system dumps these fluids into the veins, and the veins deliver them back into the cardiovascular system. What are the roles of blood? Well, a few different roles. Number one is they transport all the good stuff. You might say, what are you talking about? What is the good stuff? Well, the good stuff is oxygen, nutrients, antibodies, things of that nature. So hence, blood has a big role in immunity, and blood can also regulate your body temperature. If you're very cold, what's going to happen is the blood is going to return from your extremities to more the axial region of your body, right? The trunk, to make sure it keeps all of your organs safe, keeps them warm enough. On the opposite hand, if it's very hot outside, blood is going to go to your extremities in order to help dissipate that heat, right? So the heat will leave your extremities. So blood has many roles. Okay, let's turn our attention to what is the composition of human blood. So if you withdraw some blood from a patient and you put it into a centrifuge tube, then you centrifuge it, what you're going to do is you're going to separate the the components of the blood based on the density of those components. And so if we do that, we see three different layers that emerge. The layer that is least dense is called the plasma. This is really the liquid portion of the blood that has salts in it, things of that nature. The layer right below it is something called the buffy coat. And what that is it's a little more dense than the plasma, and it contains white blood cells such as the leukocytes, as well as clotting factors like platelets. Finally, if you look at the bottom here, you'll see the most dense component are the erythrocytes, and these are your red blood cells, and they carry oxygen throughout the body. The bottom two layers, the buffy coat and the erythrocytes, these form something called the formed elements. If we talk about the human body, about 8% of the mass is blood, and that equates to about 5 liters in volume or so. Also, if we go back to those erythrocytes, what we'll notice is that basically the percentage of your whole blood that is composed of erythrocytes is called the hematocrit, or the blood fraction. That's the word we use for that. Okay, let's zoom in a little bit more about the plasma and focus on that. So if I throw this tube up here, you want to know the three components. So think about how they centrifuge out a solution. Maybe pause the video for a second. See if you can label those three components. Okay, so let's look at those three components. The top layer is the plasma. And if we look at the plasma, what is it? Really, it is that fluid portion of the blood. It's a little sticky. It's about 90% water, has lots of salts, other ions, other nutrients, and it helps contain the waste of the blood that's eventually removed from the body. So carbon dioxide, urea, ammonia, things of that nature. Also, there are some proteins there, some of the lighter proteins in the plasma. And those proteins, if we go ahead and look to the right here, these are the three main proteins it contains. And these are the proteins. So the first is albumin. And albumin is basically something that prevents water from diffusing out of the blood vessels. So it prevents you from getting dehydrated. The second category is something called a globulin, and this includes antibodies and other proteins. Finally, we have something called a fibrinogen, and fibrinogen is something that's involved in clotting of blood along with the platelets. So these are the three main proteins that you see. You might have heard the word serum before, and you might think, what is serum? It's something we use in cell culture a lot, and then you hear it in other aspects of biology as well. What serum is the plasma, but once you take away the clotting factors. That's serum. Okay, let's turn our attention to the formed elements and look at their role in blood. So what are formed elements? As we mentioned before, they're really the blood cells, right? So the erythrocytes, the red blood cells, the leukocytes, the white blood cells, and the platelets that are involved in clotting. We use different stains to look at the formed elements. We have acidic stains, something called eosin, stains certain aspects of the cells pink. And then we have basic stains, and these basic stains, classic one is something called methylene blue, and it stains certain parts of the cell blue or purple. We're going to look at these distinct staining patterns to distinguish one formed element or one type of blood cell from another. Okay, so again, if we have that here, we say that formed elements are the white blood cells and the platelets, if we look at our tube, and the red blood cells, so the bottom two layers. The plasma is not considered one of the formed elements. Okay, and then if we want to get a little snotty about it, we could say, okay, we're using cells to describe all these different formed elements. And some of them really are not cells if we want to be picky. Just want to point that out so I don't confuse you, but we use the word cell anyway. But which ones are technically not cells? Well, the erythrocytes, the red blood cells, they don't have a nucleus. What other one? The platelets, they don't have a nucleus either. And in fact, platelets are just like just an aggregation of cell membrane, basically. That's what they are. That's one reason both of these are not cells. Another reason is they don't divide to reproduce more of their own kind. So erythrocytes do not divide to produce more erythrocytes. Platelets do not divide to produce more platelets like normal cells do. But rather both of these are produced from the bone marrow. So that's why they're really technically not cells, though you'll hear myself and many, many other people refer to them as cells. Okay, let's look at this stain a little bit more, and it's something called a Wright stain. Let's look about how we use this. Okay, so here's a smear of blood on a slide. This is our field of view. And you'll see a lot of pink, a lot of bluish purple. This is something where we've done a Wright stain on this blood sample. I want to point out some components. So almost all the cells you see here are erythrocytes. And erythrocytes are those red blood cells. They're very nice because you'll notice they're very uniform in size. And these are the dimensions that they have. If you look at a top view, you'll see that they have an indentation at the top. I put an X there. It looks sort of like a donut, and that helps increase the surface area to allow erythrocytes to absorb more oxygen. It's a really nice aspect that they have. Also, if you ever watch sort of like, I don't know, like a CSI or like a detective show on TV, you might note if there's a murder scene that they'll put a quarter down or some dollar bill or something to give you a scale when they snap a picture of the scene. Erythrocytes are really used in that aspect in biology a lot. And the reason is they're so uniform in size that if you have a erythrocyte in your field of view, you know that diameter, for example, is going to be right on 7.5 micrometers. So it's really a nice thing to use for that aspect of it. Some other odds and ends about erythrocytes you want to make sure that you know. We covered most of these here, so to put them up there. Here's some other ones that we haven't covered yet. And so what this is saying is that really they have hemoglobin, and each hemoglobin molecule bears about 4 oxygen molecules. And this is why erythrocytes are really nice for carrying oxygen throughout the body. They have that really interesting sort of color to them as the iron atoms that they contain are oxidized. So it gives blood sort of that red color. Okay, let's look at some of the less prevalent types of blood cells to see what they're all about here. So next category are the platelets here, and you can see little fragments of cell membrane that are used to help in clotting. They have an outer blue stain, and then they have a purple inner stain. A little hard to see on this picture, but if you look closely, I think you'll see it. And really how they help in clotting is that when you have a wound, say you have a cut on your hand, you're going to have exposed collagen fibers there, and what's going to happen is that those platelets are going to bind to those collagen fibers and help in clotting. So it's a really nice thing that helps there. Okay, all these other ones here are white blood cells at the bottom. Monocyte, lymphocyte, neutrophil, eosinophil. These are all white blood cells, and we abbreviate those WBC, right? White blood cells, add an S if you want to make a plural. The slide's getting a little cluttered here. Let's go to the next slide to talk about these white blood cells. Okay, so how do we distinguish these different white blood cells from each other, and what are their roles? So there's 5 that we're going to talk about, and there's 2 main categories. One of the categories is something called a granulocyte. And if you see any of this pink sort of granular substance in the background of these cells, and the middle one shows it very nicely, you can see it a little bit here and a little bit in here, then it's a granulocyte. If that's absent, then you have an agranulocyte. Let's zoom in on the granulocytes, and we'll see 3 different kinds. So the first one we see is something called a neutrophil. And we know it's a neutrophil, and the black text here is going to show you sort of the structure of each of these cells. I'll have red little pop-ups of text that tell you their function, so you can see the difference. So neutrophil is multi-lobed for its nucleus. You might say, how do we know it's multi-lobed? And the reason is we have multiple little lobes to the nuclei, and I'm putting little X's over them now. That's how we know. What's the function? It destroys bacteria through phagocytosis, right? Through engulfing those bacteria. The next thing is called an eosinophil, and this has a bilobe nucleus. I'll put X over the bilobe, the 2 lobes. That's how we know it's an eosinophil. Another reason is it's a very, very nice reddish pink stain in the background for the granules, so it really pops. That's how you know it's an eosinophil. What's the function of those? It turns off allergic responses and kills parasites. The third category of the granulocytes is something called a basophil. This has a bilobe nucleus too, just like the previous one, but a little hard to see, so that's not what I use to tell a basophil. But rather you can see the entire cell has a very sort of dark purplish black cytoplasmic granular stain. So I think it just looks very different in that regard, that it's all sort of homogeneous looking and it's all very dark. So that tells you it's a basophil. And they release histamine and other mediator factors involved in inflammation. Okay, let's turn our attention to the agranulocytes. The first one is the lymphocyte. And the lymphocyte has a very large spherical nucleus like this. It takes up almost the whole cell. That's how you know. And then what they're involved in is they help mount an immune system response. There's 2 different subcategories. There's T cells that have a direct attack, and then there's B cells that sort of have like a more indirect attack. Okay. The final category is something called a monocyte. And the nucleus here is described either as a kidney-shaped or like a horseshoe-shaped. And this is the dead giveaway. It's a monocyte. I'm going to sort of trace it here so you can see. So that's our horseshoe-shaped nucleus. Okay. What do? Phagocytosis. And the way they work is sort of interesting. When they're in the bloodstream, and I would write this down, as you're writing everything down, obviously, right? But I would really write this down. As they're in the bloodstream, what happens is they are called a monocyte. When they exit the bloodstream and go into a tissue, then we call them something called a macrophage. And that's where they undergo that, or they execute that phagocytosis to destroy foreign invaders. Okay, when we're talking about the cells, you want to think, which of these do we have the most of? Which of these do we have the least of? And there's a nice little mnemonic that we use, and I stole this from a text, so I can't take credit for this. But if you remember this phrase, never let monkeys eat bananas. The never at the top tells you the cell that we have the most of in our blood. N goes with the N in neutrophil, right? So N in never goes with the N in neutrophil. So we're saying neutrophils are the most prevalent in our bloodstream. If we go all the way down to B, you could say B goes with basophil, and that's the least prevalent, and then everything else in the middle. So remember that phrase, never let monkeys eat bananas, and it tells you the order of prevalence that these different cells occur in the bloodstream. Okay, so that was our lecture on blood. Remember, we focused on 2 components here. We focused on the composition, we focused on something called formed elements, and as always, before you proceed to the next lecture, please make sure that you're able to execute those learning objectives.